Hello, a warm welcome to you all from the Royal Society of Edinburgh for this event, The Quest to Cure Parkinson's. My name is David Price. I'm Professor of Developmental Neurobiology at the University of Edinburgh, and my research is on how our brains develop. I'm the chair for today's session. This event is part of the RSE's Curious programme, which is running up to the 17th of September, so there's still some events that you can sign up for. This offers insights from some of the world's leading experts on an array of topics addressing the theme of under the surface. Please visit the website www.rsecurious.com for more details. Okay, today the event asks, how do we find a cure for Parkinson's disease? Parkinson's affects around 1 in 375 of us across Scotland. Current treatments have not changed in over 60 years and only treat the symptoms, not their underlying causes. Today, we will hear from scientists and clinicians at the University of Dundee who are aiming to change that with the assistance of those living with Parkinson's. So I'm going to introduce the speakers to you now. We have today Dr. Esther Samler is a clinical program leader in the School of Medicine, University of Dundee. Brendan Horden is the chair of Dundee Research Interest Group and was diagnosed with Parkinson's disease in 2017. Dr. Xingyu Liu is a Marie Curie Fellow at the University of Dundee, and Professor Miratul Mukit is Professor of Experimental Neurology at the University of Dundee. So I'm now going to hand over to the speakers to give their presentations, and I will return later for the question and answer session.
Okay, thank you so much, David, for hosting this event, and thank you very much for everybody who's joined online. So I think I'm starting. So my name is Esther Samler. I am a clinician scientist and Scottish Senior Clinical Academic Fellow here at the University of Dundee. As a clinician, I'm a consultant neurologist. I see people with movement disorders, including Parkinson's disease, and I'm also the principal investigator on a large range of clinical trials in Parkinson's disease. As a scientist, I investigate what goes wrong in Parkinson's disease on a molecular level, mainly using clinical samples from patients with Parkinson's disease. I think it is really important to emphasize that Parkinson's disease is the fastest growing neurological disease worldwide, second only to Alzheimer's disease. 1 in 37 people will develop the condition at some point during their lifetime. So, for example, in Scotland, there are over 12,000 people living with Parkinson's disease. So Parkinson's disease, despite all the advances that have been made over the last decades, is still poorly understood and under-researched. So for example, as a clinician, we don't have a test for clinical diagnosis. So sometimes people visit multiple doctors and it can take years to get the correct diagnosis. And by the time the hallmarks of clinical symptoms, tremor, stiffness, slowness of movements, gait abnormalities, but also non-motor symptoms, but especially with regards to motor symptoms, by the time these symptoms, which are needed for the diagnosis, develop, 60 to 80% of the critical neurons in the brain that produce dopamine have already degenerated. In terms of treatments, what we have available, I think David already alluded to this. So the best available possible symptomatic treatment has been around since Lance Armstrong set foot on the moon. And I think that is really not good enough if you compare to other medical specialties. And in order to tackle the emerging crisis of Parkinson's disease and really offer our patients in clinic something better than just symptomatic treatment, a lot of more research and basic science needs to be done. And I think that is really like the key for moving forward. And there's a track record of this collaborative, open science that is spanning the globe with really starting from fundamental mechanisms towards treatments. That's the key for moving forward. And I think one of the things what we are extremely proud here in Dundee is all of our research relating to one of the main genetic causes of Parkinson's disease, which is the leucine-rich repeat kinase 2, in short LRK2. So it's now been about 20 years, 19 years, that LRK2, genetic changes in LRK2 have been discovered as a cause for Parkinson's disease. And why is this important? This is important because it gives us a starting point, rather than a needle in a haystack, to start unraveling what goes wrong in a cell of a person with Parkinson's disease. It gives us an opportunity to really study molecular mechanisms in cellular systems and other models to then see how this can be applied to the human system. And there has been a lot of progress here. So since 2004, when the gene was discovered as a cause for Parkinson's disease, so a lot of research that was actually done here in Dundee by Dario Alessi, who is the director of the MRC protein phosphorylation and ubiquitination unit. So he has really unraveled the pathway in the signaling of what goes wrong in a cell when LRK2 cell signaling misfires. And I think that is really like the backbone of the work that has led to us having new clinical targets under evaluation in clinical trials. Thank you.
I go next.
Mm-hmm. So I hand over to...
my name is Brendan Hawden, and I'm the person here who has Parkinson's. And therefore, I'm not going to talk about the technical part of the solution to be found in Parkinson's. I was diagnosed in 2017. I should say I was misdiagnosed in 2016, where my GP doctor thought I had carpal tunnel syndrome. As it transpired, after a lot of investigation and visiting a neurologist in Belgium where I lived, I discovered that I had all the right symptoms that corresponded to Parkinson's. Now, two things happened after that. One was that she wrote me a document saying I have a high probability of having Parkinson's. And I thought that was very peculiar, because why could she not say I had Parkinson's in a document I was about to show my employer? And I discovered that there was no definitive test for Parkinson's. And this is the reason that she gave me this document, which I was supposed to show to my employer for health insurance reasons, that it said that I had a high probability of having the condition. Then when I showed it to my employer, they wrote back to me saying, we have now accepted fully that you have Parkinson's. By the way, this certificate only lasts 5 years. And I thought, oh my God, there could be a possibility I am cured, because they've only given me a document that says that they will fund me for consultations for 5 years. I was absolutely wrong about that. I discovered very rapidly afterwards that there was no cure for Parkinson's, or no known cure for Parkinson's, and that I decided to go into this mode of self-denial. I didn't want to know anything about the condition. I more or less went through a phase of not trying to discuss it at all with people, just trying to live a normal life as far as possible. When I came to retire, when I retired here in St Andrews, which is very close by to Dundee, I came to know Esther, who became my neurologist here. And in my conversations with her, I actually developed enough confidence to be able to start finding out more about Parkinson's. And there was this book called Ending Parkinson's, which is the well-known book written by Ray Dorsey et al. And I used that book as my basis for trying to understand what was wrong with my body and what chances there were for the future. Through that particular avenue of having read the book and talked to Esther, I found out that she was involved through my wife as well, that with a group called the Dundee Research Interest Group, which was a group of people who were made up of Parkinson's sufferers, plus carers, plus researchers and clinicians as well. And we have here Esther and Myrtu, who are both clinicians and researchers. And that was quite an amazing breakthrough from my perspective of trying to understand the issue, because they both had the clinical experience of dealing with people with Parkinson's, but also the research side of things, which is an amazing benefit to have, bringing it to a group of people who are suffering from the condition. This particular DRIG, so-called DRIG, Dundee Research Interest Group, was specialized as a forum for exchange on the condition, and we found out lots of things about it. We got involved with some of the clinical trials that Esther already referred to, and we actually got to meet famous people as well. In fact, we actually got to meet a Nobel Prize winner in some of our work when we were working within the context of this forum. I think that we've gone through a lot of cycles in the forum itself. One of the cycles is trying to understand the language of researchers, because we rapidly found out that us who were the lay people who were living with Parkinson's rapidly did not understand what the science was about. It was a very complex language, and so we set a whole process in motion where we tried to understand what words the meaning to have certain words when it came to discussing Parkinson's. And we had a system where we paired a person with Parkinson's with a researcher or clinician, and tried to make sure that what they were presenting to the public was understandable by using simpler words in the presentation, or suggesting simpler words for the condition. It leaves us with a series of events that we've got planned as well. Later in the year, we hope that we will bring people with Parkinson's into the lab to show them the work that's been done here in Dundee, and as well as that, to try and make sure that the presentations are done in a way that's understandable by all. Maybe I'll leave it there for the moment, but one of the big issues that we were always grappling with is trying to find the cause of Parkinson's. In my own personal case, I don't know how it happened. I'm a bit like Michael J. Fox. I went back to my past to try and see what was I exposed to, what chemical was I exposed to that could have led to me to having Parkinson's. I don't know, but one thing I discovered when I went through my memory banks to try and see what could have caused it was that I discovered that I had lost my sense of smell. This was through notes I'd been written about by personal notes I'd written in the past, that I had lost my sense of smell in the year 2000. And therefore I feel that even though I was fully diagnosed in 2017, I had the condition from the year 2000 when I lost my sense of smell. And losing your sense of smell is one of the precursors to developing this condition. So I've had it a long time, and like Esther says, I probably lost a lot of my neurons that create dopamine, and therefore it puts the whole issue of finding a cure into a far more complex environment than you would imagine. Maybe I'll stop there. And
So thanks for organizing this event. My name is Xin Gui. I'm a postdoc fellow at Alex Chuli's group here at the Center for Target Protein Degradation, University of Dundee. So I'm a medicinal chemist by training, which means my mission is basically to discover small molecules for treating diseases, including Parkinson's disease for sure. So I'm fortunate enough. So in the past 3 years, I've been working on discovering protect molecules for degrading LRRK2, which, as Esther has mentioned, is one of the most promising target for Parkinson's disease. So we managed to discover the first RAC2 degrader in the scientific community. So it can degrade RAC2 very efficiently in the cell. So I think the challenges of finding a treatment for Parkinson's disease lies in the complexity of the disease itself. So we really need scientific innovation for sure to discover a cure for this disease. So So currently, the targeted protein degradation is one of the most popular and emerging drug discovery strategy in the field for treating diseases. So I think there are a lot of protein degraders for targets of Parkinson's disease have already emerged. So I'm very positive about those researches.
So maybe I will thank you, Xin-Louis. So first of all, it's an honour for me and my colleagues here to be addressing you. And thanks very much to the Royal Society of Edinburgh for inviting us to talk about our research background and interests and how in Dundee, not only the science, but as Brendan alluded to, as scientists and clinicians, we're working with people affected by this condition. And together, collectively, that is how we think true progress will be made. You've heard from Esther Sambler and also Xin-Louis that really Dundee has become extremely well known for basic science of Parkinson's, and particularly understanding what is occurring deep inside the brain and deep inside individual brain cells, and what is it that goes wrong to lead to people developing loss of these cells, and ultimately that causes symptoms. I think it would be, maybe the audience may be familiar with this, but and Xin-Louis has suggested this, that to understand a complex problem, and Parkinson's is an extreme, it's one of, I think, understanding brain diseases and degenerative diseases of the brain, these are the really most challenging questions in biology and in life sciences and in neuroscience. And there are different approaches that one could take to understand that problem. I think where we are now in 2023 is very much underpinned by previous research, previous advances. And I think we've reached an exciting phase. We understand very well now in Parkinson's, for example, which part of the brain is affected. We understand very well, if a pathologist were to look into the brain, which brain cells are affected. And in fact, 100 years ago, literally 100 years ago, in Germany, there was a new sort of era of science in which you could look into the brain using microscopes. They could see within brain cells of people with Parkinson's the formation of aggregations and deposits that were in people with Parkinson's and not in those who had died of other causes. And so, as Xin-Louis has highlighted, technologies are very important to understand this problem. So I started my life as a clinician, and for me, the most important thing is to try and bring to the clinic how understandings that will then lead to new ways to diagnose and treat. And I saw 20 years ago a pathway potentially to do that. And as Esther has highlighted, probably the biggest breakthrough in Parkinson's has been the identification of genes in people with Parkinson's. So most people with Parkinson's develop it, and we don't understand why. But in a very small number of people, 5% of people, they inherit a genetic cause. And we in Dundee believe that by studying why those genetic causes lead to Parkinson's could be very informative for everybody with Parkinson's. Now, why do we think that? So for me, I think the best analogy is in the cancer field. 40 years ago, we didn't have a good understanding inside cells of how a cancer developed. And that largely came from early studies in the 1980s, where they discovered genes that would cause cancers in humans. And then it took about 20, 30 years of studying how those gene pathways operate that led to now, for example, you probably see now every year at least a dozen new treatments for cancer based on understanding of those gene pathways. So in Dundee, that is very much the blueprint for how we think we can help find a cure for the disease. And so based on that sort of blueprint, we have been particularly interested in a class of gene that makes, we call them proteins. another way to think of them are nano machines. So within all of our cells, we have these tiny little machines that are performing very important functions in the brain and in your brain cells. And so a particular expertise that we have in Dundee are machines that are called kinases. These are machines that actually work as enzymes to speed up certain types of reaction. And it turns out in Parkinson's, there are a number of genes which make these kinase machines. So one of these has been the LRK2 gene. And I think this, and Esther has mentioned this, thanks a lot to the work of our very esteemed colleague Dario Alessi. There's been tremendous advances in his laboratory, which have led to how the LRK2 kinase becomes dysfunctional in people with Parkinson's. And just to sort of summarize that work, what his work has shown is that in our cells, we have to be able to get rid of damaged products and damaged components. And that's all done in a part of the cell called the lysosome. It's sort of like what we regard as being the dustbin of the cell. And if you can't get rid of the damaged materials in your cell, then that can build up and lead to cell vulnerability and loss. And Dario Alessi's work, and now Esther is following this work up in terms of the clinical trials, has found that the LRK2 pathway ultimately is trying to make sure that damage that accumulates in brain cells is efficiently got rid of in the lysosome. And that has led to now this really, really new designer-based trial, which Esther is now leading, and Dundee is one of the sites, along with other sites in the world, for patients that have treatments against this LRK2. In my own laboratory, our work has been involved with a different machine called PINK1. It's another kinase machine. But just to sort of, again, use another analogy, all our brain cells need batteries to provide all the energy for all of the processes. And our work has shown that if you can't make competent levels of PINK1, then we can't repair our batteries and keep our batteries intact through our lifetime. And ultimately that will lead to cell vulnerabilities. And we wouldn't actually understand things about the batteries of our cell or the dustbins of our cells without doing this very, very detailed, in-depth basic science. And so I think that's where we think in Dundee, we want to basically find new ways to diagnose and treat, and that's through basic science. Now, we don't work in a silo. So in our laboratories, we work, as Brendan alluded to, we work with patients, but we also in Dundee are connected with many, many companies around the world. So there are many drug companies that want to develop treatments. It's a hard business, you know. Now they can develop treatments for cancer, but as you might have read about Alzheimer's, there were many failures before there were, in the last year or so, some glimmers of success. And likewise, Parkinson's is going to be hard, but we are in Dundee are very much networked in to some of the leading companies, and it's great actually to see big pharmaceutical companies taking interest in Parkinson's. We're also very connected to all other leading clinical centres. So Esther, in her busy week, has often to travel to all the major centres in Europe for her research. The United States, we're very connected to all the big centres in the UK, including London, Oxford, and Edinburgh. And we see actually that whilst the basic research is important, we constantly have to take that knowledge and cross-reference it to what the patients are telling us, to what the clinicians are telling us, to what drug companies are telling us. And only if working together in an open way can we ultimately make progress. So I think we have a very unique way of doing science in Dundee. It's very much based on collaboration and openness. That is actually, in some ways, often not how scientists have pursued things. They've often done this in a more secretive, competitive way. But this is such a complex problem. We think that we have to be open and collaborative. And that's very much one of the hallmarks. And so ultimately, you know, the four of us sitting here all represent little pieces of a very large body of people that together we think can make progress. And so thank you again for your attention.
Well, thank you all very much for those fascinating insights. We have some questions that have come in during your presentations, and hopefully there'll be more to follow. But we have quite a few here already. So I'm going to start off with one from one of our attendees who is anonymous. And this came in during your talk, Esther, but I think it's for anyone to answer. And it refers to the LRK2 gene. And they're asking, can people therefore be tested for the gene?
I can answer this. So LRK2 is not the only gene that is implicated in Parkinson's disease, but it is one of the most frequently implicated genes. So 1 to 4% of people with Parkinson's disease will have a genetic change in LRK2, maybe even without knowing it, and sometimes even without having a family history of it. But this, this is worldwide, but there is quite a lot of variation with regards to population specificity. There are some parts in the world and populations where LRK2 mutations are much more frequent. For example, when you look at 100 people with Parkinson's disease, up to 40 people of these 100 will carry the LRK2 gene. At the moment, genetic testing for Parkinson's disease is not really embedded in NHS clinical care for Parkinson's disease, only in selected cases. If you are, for example, from Ashkenazi Jewish background, so that is where these genetic changes in LRK2 are also quite frequent. If you have a family history, like a dominant family history of Parkinson's disease, or, and that's probably more applicable to other genes, so what Miratol is working on, if you have particularly young onset Parkinson's disease. So, and that is where some NHS centers, there's also quite a lot of variability across the UK, across Scotland, across the world, but some centers do offer genetic testing for this. However, on the research side, there's a huge drive with the GP2 movement funded by ASAP, Aligning Basic Science with Parkinson's Disease, to really unravel the architecture, the genetic architecture of Parkinson's disease. So hundreds of thousands of people are being tested really in a much larger fashion, so that we get a much better understanding of the genetics of Parkinson's. And I think if people are interested in getting tested, they need to somehow see what center is close to them. And there are certainly lots of clinical observational studies. We have some here in Dundee. There are other studies in other centers in other parts of the UK and in the world and in Scotland. And it would just be a matter of speaking to their clinical research team or their consultant neurologist.
Thank you. So Fiona Newton asks, does Parkinson's disease occur at a higher level in Scotland than the rest of the UK? How does it compare with other countries?
I can also answer this. So not necessarily. I think Parkinson's disease is a worldwide problem. And while we don't really have, epidemiological data and prevalence data that's absolutely accurate, we actually think that there are 9 to 10 million people with Parkinson's disease worldwide. And I just picked Scotland because we are based here, but the prevalence of Parkinson's is the same in Scotland and the UK and the rest of the Western world. Okay,
thank you. I just wanted to raise a question from Alison White. And actually, I've heard of this as well, that there was a lady who could tell by smelling the sweat of Parkinson's, Parkinsonism sufferers, is the way she puts it. Has any further work been done on this?
Perhaps that I can answer that question. So indeed, that's a very pertinent question to Scotland, because this is very much a Scottish discovery. So in Perth, which is very close to where we're sitting now, there's a remarkable lady called Joy Milne. So Joy was Joy's husband, unfortunately, had Parkinson's for many years, Les, and unfortunately now has passed away. But after he passed away, Joy had mentioned to a colleague researcher of ours at Edinburgh called Tilo Kunath, that she could smell Parkinson's and had this sort of very, very highly specific and selective ability to pick this up, perhaps in Les before his symptoms were even advanced. And so she did a study with Tilo initially where they took some clothes from people with Parkinson's, and Joy was asked to see if she could tell which patients were the clothes from Parkinson's and which were those that didn't. And she was almost 100% correct. And remarkably, the one T-shirt that she thought was Parkinson's, but the person at the time hadn't had symptoms, turned out that person then went on to get the Parkinson's. So this was the initial discovery. And in terms of your question, what's happened since? So now Joy is now on the faculty of the University of Manchester. She's an honorary faculty member there, and she's been collaborating with a very talented scientist called Perdita Barron, who's been using this technology of mass spectrometry. And so Joy and Perdita Barron now have been able to identify. So what Joy is smelling are particular chemical signatures that are arising from your sweat. And they've been able to now break this down into what are the actual nuts and bolts of that chemical signature. And they've now published many papers on this area of research. And it's looking very promising that this could be, in the future, a diagnostic test for Parkinson's. And Joy is actually also very closely associated with our Dundee Research Interest Group, and is actually not just in this one particular project, I think is a fantastic ambassador for Parkinson's, really inspiring how she wants to help the field with her amazing ability, but also generally as a advocate to get more interest and investment in this area. Mm. So I hope I've answered that question, and you can find out more from online.
Yes, I mean, actually, this is generating quite a lot of interest. Another person has asked, if there are more people who can smell Parkinson's, would it be helpful if they reached out to someone in the research community? And if so, who?
Yeah, so this came up. Maybe I can just follow this up. So I've also encountered other people like Joy in my practice. I think that there are people who have this remarkable, our ability to smell is determined by your panoply of smell receptors. And there are these people that have these super smelling traits. My understanding at the moment is, based on the work that's now been done by Perdita Barron, they now have quite now defined chemical signatures. So as far as I know, I don't think they're looking for more human volunteers, but certainly Joy's details will be available, and they could reach out to Joy to perhaps ask if they wanted to help in that. And
then Julie Mervis has raised an interesting question, I think. Maybe dogs can be used for this as well.
So again, that's absolutely pertinent, because so that has been also exploited very successfully. So dogs have remarkable abilities to smell. And I think there has been, and this is not my research, this is Joy and Perdita's research. I think there has been work. I think Tilo Kunath has also been involved in this. And he really is, I have to actually say, in this whole area of research, it's actually the observation of Tilo Kunath and together with Joy that really was the catalyst. But I think there has been some work with super sniffing dogs in this area as well.
Okay, thank you. I'm going to shift gear a little bit now to ask from Robert Palau about the nomenclature and the language that we use. And as someone whose work is particularly related to conditions such as autism, this is a thing that comes up for us all the time. Can we please stop calling this Parkinson's disease and instead call it Parkinson's syndrome? This would better reflect the fact that symptoms vary from case to case. It would also assist both patients and their friends, families, and co-workers, etc.
I can take this question and then maybe lead over to Brendan. I mean, there's an ongoing discussion about this, and I think this is really, really, really important. And we will only be able, to resolve this question in the discussion and discourse with people affected. I think, there is a lot of discussion around, how do you refer to a person or people, people living with lived experience of Parkinson's disease or living with Parkinson's disease, because you don't want to be defined by a disease. So you don't want to be called a patient, or you don't want to be defined by your Parkinson's disease, because there's so much more to a person. So I think I think that is really, really, really important. And also what the what the person referred to is that people with Parkinson's disease are so different from each other. So if you know one person with Parkinson's disease or with Parkinson's syndrome, you know one person with Parkinson's. The age of onset, the rate of progression, the clinical presentation in terms of motor symptoms, non-motor symptoms, how functional somebody is, how somebody can engage with the environment, really differs from person to person. So I probably have to somehow pass this question on whether we should call it Parkinson's disease or Parkinson's syndrome to maybe Brendan, and I'd also be quite interested to hear what you guys have to say.
Yes. I was recently at the Barcelona conference on the World Parkinson Congress on Parkinson's disease, and I met there were 800 different people there who were living with Parkinson's, and everybody was talking to each other, but we all had different symptoms and a range of different symptoms, and we were all progressing at a different rate. We all had different doses of levodopa that we were taking. And I think it was more akin to having a syndrome rather than having a disease itself. And therefore, I would find it very easy to be able to justify changing the word disease to syndrome for that reason.
I mean, I would just add that I completely agree with what Brendan and Esther have said. I mean, in my own practice, I usually tell people that they have Parkinsonism. I mean, that's sort of a broader term reflecting actually the variability, and then later on, I try and sort of modulate that. I mean, I think what will probably help with classifications in the future are, obviously, as Brendan mentioned, 800 people, and they're all very different. Is what we can't do yet is really able to group people in to what is their subtype of Parkinson's, and that is really the next sort of frontier of classification. That will be important, for example, in clinical trials and targeted therapies and precision neurology in this area. There has been a recent breakthrough in the diagnosis of Parkinson's using spinal fluid. This has been funded by the Michael J. Fox Foundation and the Aligning Science Across Parkinson's PPMI program. They've been able to now show that you can take people with Parkinson's who look largely similar, but show in their spinal fluid that the pathological correlates are different. And there is now some discussion to take such testing and be able to then refine somebody's subtype classification. So I think at the moment, that question is, I think I agree with you. I think syndrome is probably very appropriate at the moment. And as we get better at doing the subtype identification, we can then become more accurate.
Okay, thank you. So I'm going to put two questions together here. They're really about the genetics. One is from Helga McFarlane, another from an anonymous attendee. So Helga asks, given that we have many thousands of genes, do you think over time many more may be identified to be implicated in a person's likelihood of getting Parkinson's? And the other question is Parkinson's genetically linked in any way to Huntington's, as the early symptoms can be fairly similar?
Okay, I'll take that question. So the first question is with regards to, are there many more Parkinson genes to be discovered? I think, traditionally, the first gene to be implicated to cause Parkinson's disease was alpha-synuclein in 1998. And up until then, we always knew that there was that there could be familial clustering, but it was considered not necessarily a genetic condition. And since then, there has been a constant discovery in this kind of era of gene hunting and gene discovery, very much helped by the presence of these large kindreds of families with Parkinson's disease. So I think the really common genes have been discovered. But I do think that there are many more genetic factors that either with small effect or larger effect can be implicated in Parkinson's disease. So I think in clinical practice, 5 to 6 genes are really relevant, but we have about 60 to 80 genes that can potentially cause monogenetic hereditary Parkinson's disease. And when we then look at the whole genome sequencing, these large GWAS studies where you compare lots of people with Parkinson's disease and not Parkinson's disease, we now know that there are about 90 genetic signals that may be enriched in people with Parkinson's. And you have to somehow imagine this on a scale. So you have really rare genetic variants, rare mutations with large effect. So you carry one of these genetic changes, and the probability that you then develop Parkinson's disease is quite high. And then you have lots of other smaller effect size changes that are actually quite frequent in the general population, but their effect size on its own is rather small. So you have really a whole genetic landscape of it. I do think that over the next few years, a few more genetic discoveries will be made. I think especially as all our knowledge of genetics of Parkinson's disease is mainly based on Caucasians. And I think with the effort of really unraveling genetics in non-Caucasians in Asia, India, China, that will really probably bring more genetic discoveries. The next question with regards to Huntington's disease. So Huntington's disease is really the prototype of a monogenetic disorder. So it's caused by trinucleotide repeat expansions in the Huntington gene. I think that was also discovered in the 1990s. And I think there are a couple of extremely rare causes, lookalikes of Huntington's disease that can also be caused by genetic changes in other genes. I do look as my job as a consultant neurologist. I also look after Huntington disease patients. And yes, that is correct. There can be sometimes overlap, especially, usually Huntington's disease develops in middle age, in your 30s, 40s. But there are these, extreme types where the onset is extremely young, in the early 20s. And there's a particular phenotype of Huntington's disease called Westphal variant. And that can actually present very much like Parkinson's disease in the beginning. But then, as the condition progresses, I think the whole story unravels, and the trajectory of the two conditions is then very different.
Yes, I just following. Sorry, did somebody have something to add? No. Just following on from that, Sarah Dunn asks, I'm a PhD student at Edinburgh whose father died of Lewy body dementia. I know there is a large overlap in symptoms and progression of the diseases, and was wondering if your work would impact treatment for this disease.
And perhaps I could answer that. So Lewy body dementia is mediated by very much one protein or machine or nanomachine called alpha-synuclein. So this is actually the protein that's found in deposits of people with Parkinson's, what are called Lewy bodies, discovered 100 years ago. Now, it turns out that the dementia with Lewy bodies, or Lewy body dementia, has several names. It's probably a variant. It's on the spectrum of Parkinson's, but it's much more extensive in the regions of the brain that are affected. And the rapidity, the evolution of it is a lot more faster than Parkinson's. And so it is not understood yet why it's more extensive than Parkinson's syndrome and why is it more rapid. So at the moment, I would say that the work that is done in Dundee has been mainly focusing on these kinase machines, and the links of that to the synuclein is still mysterious. I would say, though, that in the UK community, amongst movement disorder specialists, there is now very much an effort to really try and do more research on Lewy body dementia. So at the moment, there are two neurology professors in the UK who have together set up a cohort, which currently is in England, but I think it will extend to Scotland. One of them is Professor Ramona Weil at University College London. What they wish to do is to really, first of all, study many patients with dementia Lewy body, try and recruit them to their cohort studies, try and really define the clinical problems and features that they get, and then use things like brain imaging and other types of biomarker approaches to figure out how are they different from Parkinson's. So I would say that there's a lot of new activity in that area, maybe not from Dundee, but certainly in the UK. And if that person wants to reach out, I'm happy to pass on the details, for example, to Professor Ramona Weil, who we know well.
Excellent. Thank you. I'm going to jump to a question that's addressed primarily to Brendan, but maybe others can chip in as well. So Simon Stott is curious what Brendan's definition of a cure would be and what he thinks is missing from current research efforts to address this.
The cure. One of the things that we had in one of our online seminars was a discussion with Ray Dorsey. And when everybody was talking about a cure, he was saying, hold on here, a cure is a very difficult thing to manage. And when I thought about it afterwards, I think what he was referring to was that a cure has two parts. One is replacing the lost neurons that create dopamine, and the stopping of the condition was killing these neurons. So you had a by approach to the cure. Getting rid of the condition was the more challenging one, because of course there at the moment there seems to be some very good work done on stem cell replacement therapy, which is replacing the neurons that are damaged. But the actual cure, the condition itself, is something that's far more challenging. So how do I define cure? I define cure where you replace what's lost and you stop the condition. That answer the question, I hope.
That seems very clear. Does anybody else want to chip in on that? Or
I think for me, I mean, just to maybe again using a different disease as a good analogy. I mean, for me, it's the development of a drug or intervention that can arrest progression of that process. And it might be that the process is still there, but you have basically blocked progression. So the best example of that would be when I was a medical student in the 1990s, the early 90s in Edinburgh. There was this terrible wave of HIV in Edinburgh, partly because of various policies about how they were managing sort of illicit drug abuse in Edinburgh, and they'd had some policy misfirings. And so there was very large rates of HIV in Edinburgh. And at that time, HIV was very much, as everybody knew back then, pretty much a fatal disorder, which then evolved to full-blown AIDS, acquired immune deficiency syndrome. Now, with the advent of combination treatment for HIV, you have people now with HIV who every day take medication and live normal lives. If the minute they stop it, the HIV it's not gone away. It's still there, but there's no progression. So for me, it's that interruption. There are actually, interestingly, have been a few examples of HIV now being cured through various people that have had bone marrow transplants for other reasons. But nevertheless, for me, it would be some intervention that stops progression, not necessarily taking away the origin or the driver. Jean-Louis, do you want? Jean-Louis is working on the cutting edge of new approaches to cure Parkinson's using targeted protein degradation. I don't know, Jean-Louis, if you want to mention.
Yeah, sure. So thanks, Maroto, for the introduction. So we work on degrading the disease-causing proteins. For example, ARK2 is one of the targets we have been working on. So we use small molecules to get rid of the disease-causing proteins. So without the protein, probably the disease can go away in a way. So another very important protein, as Maroto has mentioned before, is alpha-synuclein. So it's also a disease. Also can cause the pathology of the Parkinson's disease. I know a lot of companies are starting working on making degraders for alpha-synuclein. That means they can remove these alpha-synuclein aggregates from the cell, so which potentially can also stop the progress of the disease potentially.
Thank you. I'm going to link together two questions, one from Vivian Kitteringham and the other from Julie Mervis. Vivian asks, what are the advantages currently of early diagnosis of Parkinsonism? And perhaps related to that, are the GPs sufficiently trained or knowledgeable about conditions such as Parkinson's?
I can maybe start here. So I think, there is a benefit for early diagnosis because usually you go and see a GP because something is not quite right. And I think getting an answer to the problem that you're experiencing is quite important. With regards to, trying to find treatments that slow down or stop the progression of Parkinson's disease, I think the belief in the field is that trying to treat people and identifying them as early as possible and starting intervention at an early time is probably much better than further advanced. So I do think there is really quite a lot of scope there. There are, in fact, clinical diagnostic criteria for Parkinson's disease that are being published by the respective bodies, the Movement Disorder Society, for example. But there are also research criteria, so to identify prodromal Parkinson's disease. And that is really, really, really important. For example, there are also worldwide several cohorts of people with conditions that we know put them at risk for Parkinson's disease. And again, we really learn quite a lot of this, following their progression, the natural progression for disease conversion, and then also hoping that we will eventually be able to stratify people, so subgroup people with Parkinson's better, and then eventually match the person with the most appropriate clinical trial or intervention. And the second part was GPs. I do think, that Parkinson's disease is a very common problem, and GPs are extremely well educated and trained to discover, and really recognize symptoms of Parkinson's disease, which would then lead to referral to a movement disorder specialist. However, it is really quite clear that even specialists sometimes get it wrong. And especially when you see someone who presents maybe out of what you would expect. So for example, if you see someone who presents with typical Parkinsonian symptoms, but at the age of 20, it's probably much higher that this person will be seeing multiple GPs. It takes time to see a specialist, and maybe because Parkinson's is not considered as the most likely diagnosis in that case. But I think for most people who develop the disease at the expected, or the condition, the syndrome at the expected age, I think if you develop shaking or or slowing down, I think GPs are very well able to pick that up and then refer to us.
Yeah, I would just follow on from Esther and say that with regard to GPs, absolutely, I agree that I think they are all familiar with the textbook and major features. In terms of early diagnosis, I'd probably put my physician hat on rather than my scientific hat. as a scientist, you could argue we should know as early as possible if somebody's on the risk trajectory for Parkinson's because of future trials that are emerging, and that they may benefit for recruitment. But the fact is, at this moment, right now, there is no treatment that can actually that can modify their disease course. So it's not like, so for example, going into so if somebody comes to see you with symptoms, it's important that you define why they have that and give them a diagnosis. But I think starting to go into more pre-symptomatic populations, one has to be a little bit more careful, because, telling someone who's 31, who's perfectly healthy and is looking forward to all the things in life, that they have potentially, very, very strong chance in the next 10 years they can develop Parkinson's, without any clear interventional option. I'm not sure that's good medicine. I don't think that's, as a physician, I'm not sure that that's knowledge that they would want to know. Obviously, if they come in a context of a strong family history and they have awareness of that and they want to know for maybe family planning purposes, that's sort of a separate question. But I wouldn't be in favor, for example, of screening really early in a very kind of more sort of sort of filtered way to make early detection, because at the moment it's not, I think, in somebody's overall well-being and interest for that information.
Thank you. So we're nearly out of time. I'm just going to squeeze in a last question. This is quite a technical question from a Dr. A. Addy. If you can manage to answer it in less than a minute, I think it would be excellent. But he asks, did I miss the bit about progressive supranuclear palsy and Parkinson plus syndromes?
I can take that question. I think we spoke a little bit about Lewy body dementia, which would also fall under the umbrella term of atypical Parkinson's disease, such as multiple system atrophy, Lewy body dementia, and then also the classical terminology of progressive supranuclear palsy. So these all have in common that they are rarer types of of syndromes with Parkinsonism, and they usually have a faster rate of progression. With regards to supranuclear palsy or progressive supranuclear palsy, there is actually a little bit of overlap and a potential link to work that we do here in Dundee. So some of the experts in PSP in the UK are Professor Hugh Morris and one of his former PhD students, Dr. Ed Jabari, who's a neurologist, both in London. And they have looked at the genetics of this supranuclear palsy, and they've identified that LRRK2, so the gene and protein that we are working on here in Dundee, is a marker, so that can differentiate people with PSP that have a faster progression than others. And actually, we have an ongoing research collaboration with the team in UCL around Hugh Morris and Ed Jabari, where we are looking at clinical samples from people with PSP and their signaling with regards to LRRK2, looking at LRRK2 kinase activity in these individuals. So that's ongoing research directly related to PSP.
Thank you very much. Our time is up. I would just like to thank all of the speakers. I would like to thank all of the attendees. I would like to remind you all to look for upcoming events in Curious. You will receive an evaluation email for feedback. I hope you found this session today interesting and informative, and I look forward to seeing you at another Curious event in the future. Goodbye.